Good. Yeah. Okay. So this is part 2 of my 3-lecture series on quantum biology, and we're still in physics, but we'll be gradually moving into biology today with some information about how you can go from simple systems to complex systems. Just to refresh your memory, we talked about some differences between classical and quantum thinking in physics, the revolution that occurred in the beginning of 20th century. And this gentleman, Schrödinger, who put everything into a very nice and elegant mathematical formulation, introducing the wave function that describes all that we know about a particle, and a Schrödinger equation, which describes the evolution of quantum particles, just like Newton's equations of motion refer to classical particles. So that's basically the situation, and at around 1930 or so, he got the Nobel Prize in 1933, and then, as I mentioned to you at the end of last lecture, he moved to Ireland to escape the Nazi occupation of his native Austria. And then he started thinking about biology and wrote this wonderful book, What is Life? But in the meantime, there are some other developments as we want to understand matter, not just at the level of a single particle or a couple of particles, or a particle in an electric field or a magnetic field. If we want to build a bridge between elementary particle physics and biology, then there's a lot of things that need to be understood. One of which is, how do we understand and describe systems of many particles, number one. Number two, many different particles. So many particles will go from several to many, and that is basically from physics to chemistry to biochemistry, biomolecules. And the second thing is temperature. How do you include temperature in all of this? Because living systems don't normally, maybe they do, but mostly they don't live at 0 Kelvin or minus 273 Celsius. So we have to incorporate temperature, which is very important in this context, and it's called statistical quantum statistics, statistical quantum mechanics, and then we'll go from there. So we talked about the properties of wave functions. There's a lot of mathematics in it, which I'll skip for in the interest of the general audience here, as I know there's only one physicist who joined us today. If any cryptophysicist is here, please reveal yourself. But basically, wave functions are, you can think about them as probability clouds or distributions with very well-defined properties that have to be integrable. Square integrable is the proper term. That is, you have to, these are complex functions in 3-dimensional space, as well as time-dependent, so they describe evolution. And they can be, which is one of the most bizarre properties of quantum mechanics, they can be superimposed or added on top of each other, as many as you want, as long as each of the contributions, each of these wave functions is of the proper type. Integrable, square integrable, and then they have to add up when you integrate them over space to the unity, so that you're not losing probabilities, you're not gaining probabilities, you're just distributing them differently. So quantum systems can be at the same time in 2 different or more, or an infinity of states, in fact. And one of these classical paradoxical situations is the Schrödinger cat. You may have heard about this experiment, the Gedanken experiment. In German means thought experiment, Gedanken, thought, where you construct a situation where the cat is in an invisible cage, and this cat is exposed to a potential poison, and poison will be released by injection if a quantum event takes place, such as radioactive decay. And until you open the cage, you have no idea whether the cat is dead or alive. So it's in a superposition. It's a probability state, and the cat is both dead and alive. And so it's one of these paradoxes of quantum mechanics that people are still talking about today. But you can also think about wave functions as vibrating strings. If you trap a particle, it'll be confined in space, and we'll have these would be kind of classical representations of quantum states in a box. Could be a first harmonic, second harmonic, nth harmonic, and all of these are good quantum states. If you think about probability distribution of a particle being somewhere, like an electron, in a we call it a square well, potential well. Okay, now we will very rapidly move on to more complex things with this lengthy introduction. That's a so-called stationary form of the Schrödinger equation, or time-independent. Time-dependent was when we had the first time derivative, when you assume that the system is in a stationary state, which could be vibrating, but it's not spreading. Then you have, this is called an eigenequation. That's the eigenenergy, and this is the Hamiltonian operator acting on the wave function. And you can solve some of these cases exactly for some potentials, like, for example, hydrogen atom. As you have the nucleus, you have electrons, and you can solve this exactly. This is what Bohr did, and obtained atomic energy levels for hydrogen. And for several other cases, you can make approximations like a helium, and that led to the spectroscopy of the day, and furthermore to molecules. But before that happens, something important is missing. Here you have the kinetic energy of the particle. This is the potential the particle feels, but that's only for one particle. And obviously in biology this is insufficient, even in chemistry. So we'll have to do something about this, because it's inadequate even for small systems. This is still talking about solving the Schrödinger equation. And then there is the question of relativistic effects, and so Dirac introduced a relativistic representation of the wave equation. Some of the particles are moving at high levels of speed, and then non-relativistic description is not adequate, it doesn't work. So this is the guy who created basically single-handedly relativistic quantum mechanics. So it's a fusion of quantum physics with special theory of relativity. A fusion of quantum physics with general theory of relativity, which is the effects of gravity at the quantum level, is still missing. After a century or so of searching, we don't have quantum theory of gravity. Dirac also introduced this very elegant representation of Bra and Ket, because it's a complex function. I will skip it, but it's a very beautiful, elegant way of solving physics problems. And he shared the 1933 Nobel Prize with Schrödinger. Okay, and this is what needs to be done to move beyond a single particle quantum mechanics. You have to add interactions, and the simplest case is for example, more than one electron filling the nucleus, and so these are Coulomb interactions, and you sum over all the electron-electron interactions that led to the entire field of atomic and molecular physics, and introduction of atomic orbitals, molecular orbitals, and eventually when you add also other nuclei, so you have a nucleus, electrons around, many electrons that interact with the nucleus, attracted to it, but they also with each other, and that changes the energy levels a bit. But then you bring two nuclei with their own electrons, and some of them may be shared, and then you form a covalent bond. Basically, you've made a molecule. Let's say two carbons is close enough, or carbon and oxygen, or carbon and two oxygens, you have CO2 and H2O, and you can start understanding chemistry, and you've created quantum chemistry. One comment, these days you hear every day, low carbon economy, carbon this, carbon that. I shudder when I hear it, because this really is so, how shall I put, moronic, I think is the best word. Carbon dioxide is not carbon, okay? So let's be scientific about it. If you want to eliminate carbon dioxide, call it carbon dioxide, not carbon. We're all made of carbon, to a large degree, by the way. So, and again, in quantum chemistry, there are two streams of development. Now you have electrons on two atoms, and some of them will be shared. And so you have to introduce a very complex wave function. Each of the electrons is represented in this wave function. It's in the form of a matrix. It's called the Hartree-Fock representation. I cannot just explain to you, the details, and probably it's not the point to give you mathematical details, just give you a general direction how science evolved. And then you can also do chemistry at the relativistic level. Believe it or not, electrons in molecules, when they spin around the nuclei, using a very metaphorical representation, they do it at a very high rate of speed. So relativity plays a role, and you need relativistic corrections to the calculations to get precise levels of energies for molecules. So that oh, sorry, this is a spelling mistake. It should be Dirac, not Dirac. Dirac, Hartree-Fock. Three names, and Dirac being a Nobel Prize winner that I mentioned to you. Okay, and that it gets more and more complicated as you start throwing in additional terms. So you have the kinetic energy of the electrons, you have the interaction of the electrons with the nuclei, electron-electron interactions, and then you have the energy level. So you have to solve this equation for all the wave functions of the contributing electrons, and eventually, well, very soon actually, you have to write complex computer code to solve it, because it's not something you can do with a piece of paper and a pencil. But what is very important at this point, when you start adding electrons, to notice what Wolfgang Pauli noticed, that and got Nobel Prize for, that electrons have spin, and that explains magnetism, as I mentioned to you, and spin 1/2, in fact, and they have a very strange property, which is called the Pauli exclusion principle. Every physical particle which has spin half integer, like 1/2, 3/2, 5/2, etc., does not tolerate any other particle at the same level, at the same state, at the same level, energy level. So it could have a different projection of spin. I'm going to be very careful now with my words, because there's a physicist in the audience and he will catch me if I say something wrong. But the Pauli exclusion principle, in a nutshell, says that particles like electrons or protons, they simply occupy a state either by themselves, one at a time, or none. There's no possibility of two electrons sitting in the same state. There are some other particles, which are called bosons, as you will see in a second, which actually have almost the opposite property. They love other particles being in the same state, but fermions don't. So in the world, there are two types of particles, fermions and bosons. Fermions are antisocial, and bosons are social, party animals. So Pauli is credited with this. And by the way, this is not a small thing, because it explains pretty much the periodic table of elements. You know, the occupation of orbitals by, the rule, atomic physics rules, how many electrons, two electrons in the 1s state, 2s the same, 2p will be 6, and so on and so forth. So it has to do with the occupation of spin states by electrons. So yes, so you have bosons, and you have fermions, like electrons and protons, anything that has a spin 1/2 or 3/2. And bosons are particles which have spin 0, 1, 2, etc. Photons, so electromagnetic radiation, from the quantum physics point of view, is a boson. It has a spin 0. And in addition, so I will not really spend a lot of time on this, but this is something that should be mentioned, because we rely on this in our everyday interactions with modern technology. an area of physics which goes in the direction of infinite number of identical, almost identical particles. So condensed matter physics, solid state physics, crystal lattices, magnets, ferroelectrics, ferroelastics, things like that led to the spawning of quasi-particles. These are not really particles, but they are quantum waves in a periodic system, such as a crystal lattice. And I mentioned to you phonons last time, so vibrations of lattices. You can think about them in the spirit of quantum mechanics as both waves and particles. So we call them quasi-particles because they are not really a particle that you can trap in a localized place, but they spread, they propagate. We'll come back to it actually in a very important connection to biology with phonons. But there are some other ones like magnons. So magnons are waves of magnetization or spin waves. So these spins are projections. Think about magnetic needles, but many of them in a row, and they basically send waves. They oscillate in space and time. Magnons, so there are some other ones, polarons, excitons, so waves of polarization, waves of dipolar excitation of electrons in spin lattices, in solid lattices, polaritons. You can couple these two. There's a whole zoology, let's put it this way, of condensed matter physics, and all of these things are called collective excitations of quasi-particles, and you can apply a lot of the same physical principles as to ordinary, not ordinary, but quantum particles. So we call them quasi. All right. So in addition to real particles, we have quasi-particles when you have systems of infinite number of atoms or states of many atoms. All right. So that's that leads us to quantum statistics, and I bring it up for a reason, because as I mentioned earlier, biology is never about a single particle. In biology, just like in condensed matter physics, we need to think about, if not infinity, uncountable numbers of players, biomolecules. It really is huge. And as I mentioned, the complication with extra complication with biology is that these are not only many, but also heterogeneous, different types. And as a scientist, you want to understand how the world is built from building blocks. So we started with a single building block, an electron or a proton, and then we made an atom. From atoms we made molecules. From molecules we can make biomolecules, larger molecules. And then we started making things like DNA or RNA, peptides and proteins. So in a way, we're building a hierarchy, right? And the question is, at what point in this hierarchy do we start losing quantum principles? Because for sure we know that at the bottom level, everything is quantum. You cannot deny it. There's no shadow of the doubt that the basic foundations of our universe are quantum mechanical. So I'm building up a story, right? So what is this graph telling you? Well, it's telling you that if you were to look at a box with molecules at some temperature, now we are bringing in temperature, which is very important as you want to go into room temperature or physiological temperature, a bit higher than that, 37 Celsius. You want to know how these particles occupy different energy states. And if you had quantum energy states equal to space, this would be a classical representation according to Boltzmann statistics, which is, if you took chemistry, you have exponential dependence on energy, e to the minus delta E over kT, so-called Arrhenius relationship, but it really comes from Boltzmann. So chemists appropriated some of the physical solutions and called them chemical, but in a way it's Boltzmann statistics. It tells you that the lowest energy state in these molecules is highest occupied, then the next one a bit less, and then less and less. Eventually, at higher energy levels, you don't have too many particles, if at all. And that speaks to the question of thermal excitation. So you can thermally excite systems of particles such that, some proportion, and I don't have a marker, but never mind. Some proportion will occupy high energy states with an exponentially vanishing probability, all right? So that's classical. But if you talk about quantum systems, and that's really important because, again, if we go to biology, we'll have to keep this in mind. The things change completely. So now it depends on whether these are fermions or bosons in this box. So if these were fermions, I told you that they don't really like any neighbors. They hate antisocial particles. So this is the lowest energy state. You can only put one particle there. Next one, next one, next one. Each state will be occupied by one, and you build up this ladder of states, and it's occupied up to, when you fit all the particles. And that's why the states of atoms, electronic states of atoms, start from 1s and 2s to 2p, 3s, 3p, 3d. You kind of occupy whatever is available gradually, right? You can ionize them with extra energy and eject some of the electrons from these orbits, but you build it from the bottom to the top. And these are fermions, electrons are fermions. So this is kind of a simplified version of the sort of atomic in the box, let's say fermions in the box. Bosons, on the other hand, and we'll talk about this gentleman, S.N. Bose, in a minute, a very interesting character. Bosons will love to have neighbors. They are social animals, so they will try to stay in the lowest energy level. Okay, so the question is, whether you're dealing with bosons or fermions. So you see, these are hallmarks of a completely different behavior. If your system of many particles, biomolecules, whatever the case may be, is classical, it'll have an exponential distribution among the energy levels. It's called thermal, a thermal distribution because it's mathematically it'll be described by something that depends on the temperature. If something is non-thermal, away from this Boltzmannian, that usually means there's something really interesting and strange going on. And that would be a hallmark of either quantum or some non-equilibrium, some energy pumping or something like this. And fermions would have the most, bizarre distribution, and bosons also, but maybe a bit less so. Okay, so now about Bose, S.N. Bose. So this is a very interesting story. To some degree, maybe still stuff like that is going on today. In 1924, so S.N. Bose was an Indian physicist. Actually, he never got a PhD. He had a master's degree from Calcutta, I think Calcutta. And he was just a self-taught polymath, a genius. And he wrote a paper that couldn't get published. And for some reason, I don't know how that happened, but Albert Einstein got hold of the paper and translated it into German. And the paper got published in the premier journal of the time, Zeitschrift für Physik. And Bose became famous. And he was famous for many other reasons. And he became a fellow of the Royal Society without a PhD, which is quite amazing, right? I mean, this is a very exclusive club. I think on this campus we used to have 5 fellows of Royal Society, but anyway, this is really a very prestigious organization, the oldest scientific society on the planet. And elitist, this guy, okay, so he was nominated for a Nobel Prize for his discovery of this Bose-Einstein, now we call Bose-Einstein. Einstein translated the paper again. So this is how lowly you can get, being Einstein and translating somebody else's paper and becoming famous by association with S.N. Bose. So it's a really funny story. And so Bose was nominated for a Nobel Prize, but rejected by a Swedish scientist, Oskar Klein, who was on the committee. Do you know how Nobel Prizes are awarded? How it goes? Okay, so just a 2-minute interruption about Nobel Prizes. So there's a committee, and that committee, you can propose to the committee names of famous, every year they gather and they look at some nomination. Who can nominate somebody for a Nobel Prize? Previous Nobel Prize winners, all professors in Scandinavian universities, so from Stockholm, Oslo, Copenhagen, as well as a select group of eminent scientists around the world who are invited to propose somebody. You cannot just send the names of some, out of the blue and hope that this will be read by anybody. But so these are the three groups of people that can propose somebody for a Nobel Prize, and some very famous people never get it, and so Bose is one of them, and Henri Poincaré is another. It's in many societies, sociology, so I don't know, there are very many explanations. In this case, somebody didn't like Bose. But I think he was asked towards the end of his life whether he was bitter about it. He said he got enough distinctions and recognition in his life and wasn't bitter at all. But what's interesting is that it took a long time. So one of the predictions of his work is that bosons confined in space will undergo something called the Bose condensation. It's actually important in biology because there were some predictions that some biological systems exhibit Bose condensation. Bose condensation means that a microscopic number of particles, a huge number, let's say, reside in the ground state. They become correlated. It's called a condensate. They form a condensate. It's a bit like looking at a population of young men between the ages of 18 and 24, and then forming an army. So an army would be this Bose condensate. They all march in lockstep, they wear uniforms, they all look the same, right? And then the rest of them are just party animals and whatever. So Bose condensation is like everybody being drafted into the army. And it took about, 70 years or so for Bose condensation to be very experimentally found. And this is the irony of it all. Einstein translated Bose, so Bose got published and became a fellow Royal Society. Did not get a Nobel Prize. That was 1924, right? So 77 years later, or 76 years later, these two gentlemen, Cornell and Wyman from the University of Colorado, observed a Bose-Einstein condensate of 2,000 rubidium atoms, and they got a Nobel Prize. Okay, so somebody who confirmed Bose's genius 77 years later got a Nobel Prize. He didn't. Luck plays a very important role in science, but they had to cool. So it wasn't easy, by the way. They had to cool it down to this. I think it's 1/1,000,000 of a degree Kelvin. So Bose-Einstein condensate is a new state of matter. It's a macroscopic quantum state where all these condensed particles share a wave function. It's a macroscopic quantum wave function. So the Schrödinger wave function becomes macroscopic. All 2,000 rubidium atoms were part of this condensate wave function. And so, but some people thought about this and they said, well, wouldn't it be nice if our brain was actually a quantum condensed wave function? Because we have a huge brain, right? Compared to electrons or protons, this is massive, 1.6 kilograms or so, but we have a coherent thought process, right? We think coherently, sometimes not, but most of the time we try to think coherently. So there are some theories or proposals that actually our brain, and I'll talk about this a little bit on Thursday, that our brain is an example of a Bose-Einstein condensate, where we don't know what is a thought, but would kind of explain some of the mysteries of the brain if this kind of quantum process took place in our brain, that a thought is a condensation of some ideas floating around, eventually manifest themselves as a unified, coherent state in the brain. So we don't have any practical applications of this yet, but maybe it is actually a way to understand how we think. And just for those of you who are mathematically inclined, the difference between the occupational probability function for bosons and fermions is just in the sign. If you have 0 here, it's the, and this is the function I wanted to write down, is basically Boltzmannian, e to the minus epsilon over kt. Epsilon is the energy, value of energy. If you have a plus, it's fermions, with a plus, minus is bosons, and 0 is classical particles. So one function covers all cases, but you just change this number in the denominator, again, minus bosons, plus fermions, 0 for classical particles. And there is actually a difference. Graphically, this is a probability distribution function for Fermi-Dirac or fermions. It's a characteristic step function. It's used a lot in semiconductor physics. This is what we use for amplification, and this is called the Fermi level, transition to conduction. All that we use today, cell phones, laptops, TV, is based on semiconductor technology, which was possible because of this Fermi-Dirac function for electrons, how they occupy energy levels at the time of temperature. And the sharp transition is at 0 Kelvin, and it becomes softer and softer as you increase the temperature. Green is at room temperature, basically, 300 Kelvin. And purple is when you kind of melt this thing. And that shows you the change as you go, again, graphically, the 3 cases. Fermi-Dirac, so fermions, bosons, and classical in between. So this is energy. Energy in terms of thermal energy, we'll come back to it, why it's important. In biology especially, thermal energy plays a very huge role because it's something that is quite sizable. And this is the probability function that you find a particle with this energy, this energy, this energy, this energy, and that's diminishing. The higher the energy, in all cases, the lower the probability of manifesting itself. So as you can see, bosons really like to be at high energies, at low energy states, and have high probability of having many of them. Fermions can never have more than one, and so on. Okay, now I'm going to say a few words about quantum theory of solids and phonons for a very specific reason, because it has a lot to do with quantum metabolism that I'll speak about a bit later. So what's this? This is actually one case. So we saw Einstein in several roles already, right? Einstein, the genius who came up with the special theory of relativity, explained the photoelectric effect, got the Nobel Prize. General relativity, didn't get a Nobel Prize for it, but he built these 2 major fields, special and general relativity. And then he translated Bose, and his name is on the Bose-Einstein condensation and Bose-Einstein distribution. And he made one mistake, at least, but the one that we know of. Around the same time, maybe in late 1930s, physicists were measuring specific heats of solids, different solids. And that means, so the question is, how much heat you have to deliver to a piece of solid to raise the temperature by 1 degree Celsius, right? And you do it at different temperatures. It turns out, so the specific heat is the amount of heat per degree, per unit of mass. And it turns out that at low temperatures, this specific heat function goes down very rapidly with the 3rd power of temperature. Nobody could explain it. Why is this? Yeah. At high temperatures, it becomes constant. It's called the sorry, I forgot the name of these. What is the high temperature limit? It's 3 and KT. There's a name attached to it, it'll come back to me. Not
Is the Debye model? Is that
Debye temperature, you're getting ahead of yourself. It's not Debye temperature. Debye was the guy who explained it. Well, we'll come to it. But I'm talking about Einstein. And Einstein assumed that this a solid is composed of atoms or molecules, all of which are vibrating at a given frequency. And you sum it up with the Boltzmann factor, and you obtain the energy of the entire solid, and then calculate the derivative with respect to temperature, get specific heat. Unfortunately, at low temperatures it breaks down. It gives you an exponential drop rather than polynomial, t to the power 3, instead of e to the minus energy over kT instead of t to the power 3. So it's wrong, and that's Einstein's mistake. However, it's not completely wrong because it applies to gases. So you can explain the specific heat of gases using Einstein's thinking, that there is, a gas of identical molecules. Each molecule has a vibrational frequency. Then you can get good results, but not solids. So Debye, as Arad mentioned, Debye explained it properly by assuming that solid is actually supporting waves of motion. So all these atoms move like waves on a sea, except the sea is a bit more chaotic. It would be very regular waves propagating from one end to the other, reflecting back and so on. And when you replace this, so you still assume Planck's quantization of energy. So energy is proportional to the frequency of the wave. And do the calculations assuming finite size of the solid, the separation between the atoms, and so on. You obtain the correct dependence. C is the specific heat or heat capacity. Specific heat of the solid goes like t to the power 3 at low temperatures to constant, gas constant R at high temperatures. And that's basically the story. And that was Einstein's model. He got the high temperatures right, the low temperatures wrong. But as I said, applies to gases but not to solids. All right, why is this important? Because in a few minutes I'll try to convince you that you can, using the same thinking, you can explain metabolic behavior of living systems. Sounds like a stretch. Actually, mathematically, it's not. I just want to emphasize one thing. By the way, Debye got a Nobel Prize as well. So as I mentioned to you, almost everybody I mentioned got a Nobel Prize, except for this poor S.N. Bose, your countryman, who was a genius, but not recognized in Stockholm. And so the Debye model. Why is this important? Because it's a statistical physics approach, quantum statistics, showing that solids manifest quantization by temperature dependence. Okay? And this is important because it's a completely unexpected result if you think about it. You look at a system of atoms behaving collectively, but the assumption underlying the Debye representation is Planck's quantization of energy, h omega, Planck's constant times the frequency. Whether it's electromagnetic frequency in the photoelectric effect, as we talked about, or it's a phonon or vibrational frequency in a solid, it's still quantized. So, we'll come to it in a few minutes, there's this obsession in the field of quantum biology that you have to see things, really exotic things like superposition, entanglement, action at a distance, tunneling, to prove that something is quantum. But you don't have to. This proved to physicists that solids are quantum, even though they can be macroscopic. If you look at the specific heat as a function of absolute temperature, this t to the power 3 is a signature of a quantization of energy of the solid. Okay, so I'll just rattle off some other microscopic quantum effects, which I think I mentioned already briefly before. So, in solids, or physical systems, so superconductivity, superfluidity, these are undeniable quantum effects occurring at microscopic sizes. You can have a piece of a superconductor, you can have a piece of, or like a bucket, or a container with superfluid. These are coherent, quantum coherent systems. Laser actually mentioned about, and now crystal vibrations or phonons. As long as the system is regular, you see it more clearly at lower temperatures, of course, because this T to the power 3, then there's an inflection point, turns over and becomes constant at high temperatures. So if you were only looking in high temperatures, and then the high depends on which solid you're talking about, and this transition is the Debye temperature that Aaron mentioned. So the Debye temperature is the temperature at which a solid becomes, for all intents and purposes, classical. Above which, below is quantum. Okay. But nonetheless, if you probe it at low enough temperature to see it, then obviously it is proof that at the bottom level you are dealing with quantum phenomena. And as I mentioned to you, magnetism at all temperatures where you have magnetization. A Curie temperature, by the way, is the temperature below which a magnet becomes ferromagnet. You have line spins. In other words, overcomes thermal fluctuations, and magnetic spins align themselves and form a magnetic domain. So quantum doesn't necessarily mean an electron interacting with another electron or proton or being hit by a photon. Quantum means a lot of other things as well. Okay, I'll just mention, so we're moving now into modern era. Janos Neumann, or John von Neumann, as he's currently known, was a brilliant, another of those, somebody said he was the last polymath, the last genius of 20th century, who wrote brilliant papers in mathematics, physics, computer science. He actually created basically computer science, information theory, statistics, topology. But he laid the foundations for quantum computing. And quantum computing is important in this context as well, because as people like Roger Penrose argue, our brains operate non-algorithmically, perhaps as quantum computers, which would explain a lot of incredible potential of the human mind. So John von Neumann laid the foundation for quantum computing, worked in operator theory, also cellular automata. I don't know if you're familiar with Mathematica, Wolfram, and this whole area of mathematics based on cellular automata, which is basically discretized, use discretized space and define operations. It's a bit like what computers do, right? Digitize everything into zeros and ones. And he also came up with cellular automata, and some people like Wolfram believe that you can replace all of mathematics by cellular automata. You don't have to write differential equations. In other words, continuum mathematics can be replaced by discrete mathematics. And if you go further, you can actually conclude that all of the universe is a big computer which just changes bits of information, zeros to ones, and all of this is somewhere in the ether. I'll come to it actually in a minute, why this is such a powerful concept, whether we should be thinking about the universe as a digital computer or not. Okay, and this is perhaps one of the greatest American physicists of 20th century, Richard Feynman. I mentioned his name before. He laid the foundation for nanotechnology, which is, again, another area very closely associated with quantum biology, because at the bottom level, as you will see in a second, biology is especially cell biology and molecular biology, is nanobiotechnology. We're looking at the nanometers spatial dimensions for things like DNA, RNA, proteins, and nanosecond transitions. And this is kind of this somewhat blurry no man's land of physics. Not anymore, but at that time anyway. This was 1960 something, no, 59 actually. During the annual meeting of the APS, American Physical Society, at Caltech. He was from Caltech. He said that at the atomic level we have new kinds of forces, new kinds of possibilities, new kinds of effects. He was talking about, and he called it the loss of room at the bottom. Nanotechnology, basically, the nanosystems have new rules which are kind of in between classical and quantum. This could, so repeating chemical forces repeatedly can produce weird effects, one of which is the author. So see, this is he's alluding to biology being really nanophysics, right? Repetitive forces at the nanoscale produce weird effects, including the author. In other words, a living human being. So, of course, so he was talking about maneuvering things atom by atom in 1959, manipulating systems such that you can move one atom at a time. And by the way, do you know who on this campus has the world record of maneuvering a single atom at a time?
Hegman?
No, close.
Wolkow, Professor Robert
Yes,
Wolkow?
yes. Bob Wolkow in physics department is in the Guinness Book of World Records. He established technology here that allows movement of a single atom. And I think one of his postdocs created the University of Alberta logo by moving atoms. Look it up. So, but Feynman predicted this in 1959. That alone, the power of the human mind is quite amazing. Okay, so now we're shifting gears and finally getting into the conversation about quantum mechanics and life. So we have, unfortunately, in biology, and please correct me if I'm wrong, but there's only one theory. That's Darwinian theory of evolution, and it's very old and long in the tooth. I think it needs replacing or augmenting or something. I juxtaposed it with Schrödinger, the Schrödinger equation, which already is a little bit obsolete, but you know, very general anyway. So there's, where does quantum weirdness fit in? Quantum effects would explain a lot of biology, one of which is how come biological systems, which are so heterogeneous and so hierarchical and so diverse, operating at different scales of dimensions in space and time, maintain their coherence. They maintain the unitary sense of self or purpose. This is one of those big ideas that require deep thinking and explanation. But the hope, to me at least, is that life on this planet, which has been going on for 2 billion years, it's an ongoing single experiment, by the way. Right? To our knowledge, it's all started with the RNA soup, and then it all evolved from there. We don't have a separate experiment going on Mars. Maybe there was, but I don't know. But on Earth, it all started with single life forms, and then single cells, multicellular organisms, and then Darwin kicks in, and all proceeds as it should, perhaps. I don't know, with some hiccups on the way. So what I'm saying is that we have a single experiment with multiple replicates going on for 2 billion years. And therefore, and this is a search, life on this planet, you can call it a search for an optimization under various conditions, right? So living forms are diverse to start with, but then the best survival of the fittest, the best adapted to a given condition, proliferate, and then this advantage is retained for future generations. Whatever is an advantage in survival is retained. So I think this way of thinking allows you to hopefully accept the fact that if we, as humans, discovered quantum mechanics as the most fundamental law of nature, useful in making your iPods, iPhones, and cell phones, and laptops, and everything else, telecommunication, why would life forms, other than human beings, not exploit it over the past 2 billion years and uncounted trillions of replicates of these experiments, right? It's hard to believe that we are that. I'm not arrogant, by the way, and I hope I'm not. But as a physicist, sometimes you say things which you regret. So these are the gifts of quantum mechanics. If quantum mechanics, and I believe some of it at least, is being used by us as human beings, living beings, these are the gifts of quantum mechanics to nature, if nature wanted to use them. So there's delicate stability. And sometimes you hear that life is fragile, right? But it's also robust. It's both fragile and robust. So there's some element of fragility in quantum states are somewhat fragile. We can destroy them by thermal fluctuations, by collisions. But then they can form condensates and become quite robust. As I mentioned to you with Bose Einstein Condensate, with laser, also with energy supply. You can maintain, and you need to maintain life. Without energy supply, life does not exist for long. I mean, try not eating for a few days. Coherence is the most important aspect, which actually captured the imagination of biophysicists in the past. Coherence, that is this, feeling, not only feeling, but acting as a unit. And when you look at the chemical forces, chemical interactions, they're very, very slow. Diffusion driven. Diffusion is a very small process, very slow process, and cannot explain reaction times, for example, of some of the best achievers, even not best, but reasonable achievers in the human sphere. Things like being able to perfectly return a serve going at 190 miles an hour, something like 120 miles an hour, and with a reaction time which must be less than a millisecond. What basketball players achieve, what baseball players achieve, the speeds and the reaction times and the precision, some of this really defies explanation using classical terms, especially chemical reactions. I mean, come on. It has to be electromagnetic at some stage. It probably involves some quantum effects, although I cannot explain to you and give you a simple explanation why somebody like Federer can do what he does. So coherence, quantum information storage. Information storage, and on Thursday I think I'll have time to talk a little bit about the human brain, how much information we can store in the brain in principle, and how fast we can process it at how little cost. That's quite amazing, and if you add to it quantum information storage and processing, not we, but other physicists are working on quantum computers as we speak, which are becoming a reality. That's another reason to believe that Mother Nature has discovered quantum computing as well, and perhaps it's taking place in our brains as we speak. So stochasticity is this reliance on chance to some degree, and probabilities rather than certainties are part and parcel of living systems, and most of it is really trying to hedge your bets on probabilities rather than becoming machine-like robots that are completely deterministic. So living systems definitely are stochastic beings. The question is how you bootstrap probabilities to achieve end goals. For example, cell division. Cell division is a process which is incredibly error-free most of the time, but not always. There are mutations which interfere, and of course it can get derailed into things like cancer. but yet, you know, probabilities of perfect outcomes are very, very high, amazingly high. And it's because of this error correction mechanism, let's say, using the language of computer science. Quantum statistics, I mentioned to you, so again, it kind of includes both the probabilities as well as quantum effects. Tunneling we know exists in some molecular systems, quantum tunneling that is, which means reactions, some catalytic reactions taking place against all odds. From classical thinking, it would not be possible to take, they wouldn't be possible to take place, but there is quantum tunneling and enzymatic catalysis. That's one of the examples in molecular biology where quantum physics or quantum chemistry is being used. This, I leave at the end, action, this weird action at a distance that Einstein didn't believe in, which, by the way, has been demonstrated over and over again in experiments, that particles get entangled or correlated, and if you force one particle to reveal its state, the other one will do simultaneously, even if it's separated by distances that defy any interaction, physical interaction. So that means quantum entanglement. So this has been demonstrated in laboratories, in physical laboratories, over and over again. And again, if we are allowed to hypothesize or fantasize a bit, if we can think about quantum brains, and not all of, I don't know if we're all entangled here in this room, but maybe some of us may be entangled to some degree. Sometimes you're surprised by somebody basically speaking your words before you say them, right? Because you think alike, or maybe you are emotionally involved with somebody, and you discover that they were thinking exactly the same as you are, or sometimes there's a strange premonitions or dreams people experience, and very difficult to blame it all on chance. It could be something like quantum entanglement between brains of living human beings. I'm just leaving this as something for a late night discussion over a bottle of wine, but nonetheless, or beer, or Powerade, if you don't drink, whatever. You can think about it. I have to say that many examples of people reporting such strange manifestations of being connected with somebody over a large distance without really physical connection, but yet either feeling something happening or thinking the same way or something along these lines. And another interesting thing is that a lot of creative breakthroughs in science occurred almost simultaneously with more than one person coming up with the same idea. Anyway, that's just another anecdotal example. So yeah, we of course as physicists want to reduce everything to the same level. So reductionist science, of course, has its own flaws, but nonetheless it has some grain of truth. So we start with nanometer, and biomolecule or macromolecule can be reduced to its constituent atoms into nuclei, electrons, and nuclei into neutrons, and elementary particles into quarks. So, of course, the basis of all of this is quantum, but that's not the question. The question is it relevant, and is it all quantum? And my personal opinion is no, it's not all quantum, but there may be something quantum, and if it is, it could be very important. So in living systems, we are dealing with hierarchies. Unlike in physical systems, we talked about lattices, regularity, soldiers moving lock, stock, and so on, marching good step type and things of this nature. In biology, it's more like hierarchies. We can start with the elementary particles giving rise to condensed matter, this atoms, molecules give rise to chemistry, chemistry to biochemistry, and then you, through proteins, nucleic acids, sugars, you can build up a cell. But this is one kind of interesting tidbit of information I want you to think about for a few minutes, which basically gives us both a limitation to our understanding of the world around us, and it's a sobering thought about brute force, brute force approach to science, a reductionist approach to science, which I don't agree with. It's called the combinatorial barrier. If you can think of any system of elements and their interactions, you can come up with this 10 to the power 110 is a physical barrier to even inspecting that system ever, anywhere. So it's a hard stop on any brute force understanding of any system. And by the way, there are many systems which are that big. For example, the number of possible DNA sequences or the number of possible protein sequences far exceeds this number. Let's say amino acids, there's 20, right? So 20 amino acids, if you string 100 of them, it will be a very small protein. And if you start mutating them, then for a given amino acid, which is only, a given peptide or protein, which is only 100 amino acids long, you have 20 to the power 100, which is way bigger than that. So the space of proteins, or the space of DNA sequences, is astronomically larger than this number, okay? So if you wanted to talk about proteomics or genomics as a science that can be exploited completely, forget about it. It will never, ever be done, and the reason is here. This is not an arbitrary number. 10 to the power 110 is actually the product of the following. The number of atoms in the universe, let's say proton, the weight of the entire universe in protons multiplied by the age of the universe in picoseconds. Why is that? Because if you imagine that the entire universe is a supercomputer, with every atom in the universe being one element of that supercomputer switching states from 0 to 1 on a picosecond time scale, which is really fast, then the universe as a computer, for the entire age of the universe, 16 billion years or so, 15 of them, working non-stop, and every atom switching states, would give you that number of states that the computer would go through. You see? So if you build a supercomputer out of all the atoms of the universe that were all switching states on a picosecond time scale, which is atomic transitions, that computer would only be able to inspect, let alone do any calculations on them, this many states. So clearly, you cannot inspect all the protein states, you cannot inspect all the DNA sequences, forget about it. It's a hard stop. All right, so that's the so. Now, back to now, so today I'm going to take you through some interesting, in my opinion, connections from physics to biology. So making a transition, and then on Thursday we'll really talk about what can be quantum there. But so we start from physical objects, making them bigger and bigger, and comparing sizes. So an electron is a fraction of a femtometer, proton about a femtometer, neutron a bit more than that. Hydrogen is about half angstrom, so this is nanometers, 0.05, so half angstrom, 10 to the minus 10 angstrom. Hydrogen bond is about quarter nanometer, carbon, oxygen. Water is about 1.4 angstroms, 1.3 angstroms in size. These are the masses. So now we are at water is 18, of course, Daltons, right? And now we're going into biochemistry and biology. So amino acids about 110. An average amino acid 110 Daltons. So if you want to quickly figure out how big, what's the mass of a protein, you basically multiply by the number, 110 by, or 100, let's say, if you don't want to clutter your mind, 100 by the number of amino acids. So tubulin, which is 500, about 450 amino acids, monomer, alpha tubulin or beta tubulin, the molecular weight would be about 50 kilodaltons. Just multiply by 110, you get the mass of tubulin. If you want to do actin or any other, just do this. Nucleotides from 309 to 324 daltons. Cells, so the mass of a cell, prokaryotic, so no nucleus, is between 5 and 50 to 500 billion daltons, gigadaltons. And eukaryotic will be much bigger, from 2 to 110 to the 13th daltons. So, as you can see, we're now going dramatically up in size from physical system that we discussed before. I tried to convince you that don't worry about it because physical systems can be big in quantum, but one difference is that these are systems which are big and heterogeneous, right? So if you talk about the laser or a magnet or a crystal, these are repetitive, identical units. I'm not saying it's not possible to create a hierarchical structure which is quantum. I'm just saying we have to be careful about this. Now, so this is kind of a graphical representation of the sizes in biology. In physics, we typically don't really sweep such a range. So from, let's say, 10 to the minus 11, so that would be, yeah, 0.1 angstrom. Nothing really interesting happens, but from hydrogen atom, we angstrom, and of course 10 meters human, and giant elephant, let's say. But with DNA, nanometers, ribosome 20 by 30 nanometers, virus is 100 nanometers, E. coli cells, the smallest cell, living bacteria, 2 microns, red blood cells 2 to 5 microns, hair 0.1 millimeters and 5 millimeters, and so on. So we have to span about 11 to 12 orders of magnitude in sizes to understand biology if we want to build it from the bottom to the top. Bottom-up approach. Now, for time scales is even more difficult because, and I would even back up a bit. We talked about picoseconds. Picoseconds would be atomic vibrations, chemical bond vibration, 0.2 picoseconds actually. So 10 to the minus 2 times 10 to the minus 13s. If you look at starting from atomic processes, it would be 100 nanoseconds. By the way, 10 to the minus 7 is a very important number because this is currently what is the standard of our computer simulations in molecular dynamics, which is you can, so-called all atom simulations. You can simulate a whole protein or a protein complex for that long with this time step. Actually even smaller, femtoseconds. So we are scratching the surface in terms of understanding biological matter computationally. But here it is, okay, so MD simulations, maybe microseconds would be a world record. Diffusion, so I told you diffusion is too slow. We're talking about tens of milliseconds to milliseconds, diffusion processes. Now cognitive responses, by the way, and I don't believe this, but there were experiments done in Berkeley in the 1980s by Libet on the speed of response, human reaction time to any stimulus. Let's say, my voice, or you watch something, or you react to something. And there was a term 500 milliseconds, so half a second is the response time. But I think it's conscious. I don't believe that subconsciously we are that slow. I'm sure we can be much faster. So at any rate, human lifespan is a billion seconds. So we have, as I said, I would back it up to 10 to the minus 10, so angstrom, from atomic dimension, time scales to lifespan, we're talking about almost 20 orders of magnitude in time. So you have to build a hierarchical structure, that is, things happening on a picosecond scale, atomic scale, have to bootstrap into something which is on molecular nanoseconds, complexes on microseconds, cells maybe milliseconds, and then organisms seconds. Cell division will be hours, right? So this synchronization going on from atoms to organisms is to me the greatest mystery. How it is done, you know, how it's orchestrated and organized is the greatest mystery. How do we, and this is again on the note of quantum effects, one of the quantum effects that we know that we experience as human beings is the sensation of light, vision. Our sense of vision is a quantum apparatus which can, the eye can detect a single photon, just like in the photoelectric effect we talked about. But how is this amplified so that we perceive it as light, as a flash of light? Maybe not one, let's say even 100, and you've noticed there was a flash of light. Your brain perceived it, and you know it. Your cognition registered it. How is this? How can we span these many orders of magnitude in space and time and maintain this coherent perception? And to make it even, this is kind of another hierarchical organization where I fit different fields of science that currently explore it. So at the atomic level, of course, quantum physics, we talked about molecular quantum chemistry. Now we go into biomolecules, and there's something called quantum mechanics, molecular mechanics, which is for biomolecular systems, we use a hybrid. Most of the biomolecule, for example, an enzyme, could be classically analyzed as just balls and bonds and sticks moving around. But the receptor on that molecule, an area of, let's say, 5 to 10 angst, maybe 5, 6 angstrom radius, will be modeled and understood quantum mechanically. When it binds a ligand and triggers a catalytic transformation, it does so by a quantum tunneling process, for example. So we are here in this no man's land at this moment, mesoscale, where maybe quantum biology, which could be a combination of classical and quantum concepts, we don't know exactly where, you know, the boundaries are blurred. And then, of course, at the level of an organism, we move into thermodynamical, microscopic mechanics, Newtonian world, and so on. But yeah, that's the whole mystery. And this is how we do the modeling, by the way. We do it at the length and times currently. This is state of the art in terms of physics, modeling biological systems. Between, let's say, picoseconds and nanometer scales, we use quantum mechanical, can use quantum mechanical representation molecules. Nanosecond, micrometer, atomistic models, we forget about electronic degrees of freedom, electrons being quantum. And then we do something called the coarse grain, so further approximate, let's say, lump all the atoms of an amino acid into one and represent the whole amino acid as a point with a mass. And then go into continuum models on the time scale from milliseconds to seconds and millimeter. So you can represent the cell as a continuum, cytoplasm with some membrane boundary, etc. Okay, and I will finish, no, I still have a few minutes, but I'm almost finished for today with, so this is all to set the stage, to tell you where we fit biology into the physical thinking. Important energy scales, so we talked about length scales, time scales, hierarchies, right? And at the bottom level, we have to be very cognizant of energy, because biology does not exist without energy, constant energy input. And basically it's all about resources, nutritional resources, converting them into structure and function, organization and performance of various functions. So these are the important, and in biochemistry and biology uses, you know, kinetics, but kinetics underlies energetics. And this is a transition between KDs, you know, kinetic constants for reactions, and binding energies in kilocalories per mole, which in physics we don't use, but it's okay. So the strongest bonds, which biology, by the way, uses, but not so commonly, are covalent bonds, about 90 kilocalories per mole. These are the distances over which these bonds operate, 1.5 angstroms. Ionic, so it's electrostatics, and somewhat, how should I put it? Unfortunately, biology forgot about electrostatics. For some reason, there's very little about electrical charges, but to my mind, almost everything in biology is electrostatically driven. So ionic bonds, a bit weaker, disulfide, a bit weaker still, salt bridges, ion dipoles, 6 kilocalories per mole. And most of the action is at the hydrogen bond level, which is somewhere between 0.5 and 12 kilocalories per mole, a bit longer orientational dependence. So hydrogen bonds are flexible, reorganizable, you can use them as Lego, build structure based on that. And then van der Waals interactions, which are basically polar induced dipoles. And I want to emphasize two numbers only from that table. Maybe keep in mind, roughly speaking, that all the bonds will be somewhere between 0.5 and 100 kilocalories per mole. But these two numbers are very, very important. KT, which is thermal energy, which is free, because all of life, pretty much, at least human life, exists at physiological temperature. So at this temperature, 0.6 kilocalories per mole is for free. Just because it's warm, you get it. Every atom of your body can use this energy for free. And also, at the same time, think about it as anything below this level is noise, because it can be completely washed out by just thermal fluctuations, molecules bumping around. So 0.6 kilocalories per mole is the threshold. Anything below is noise. And then finally this, speaking to the non-equilibrium nature of life, metabolism, we need to get these two guys, GTP and ATP, especially ATP. So this is the molecule, I call it the quantum of biological energy, ATP, quantum of biological energy, because most of the processes, almost every step of every process uses one ATP molecule. Every motor protein, kinesin, dynein, myosin, every filament, actin, microtubules use GTP. To make one addition, to make one step, you need to use one molecule of either ATP or sometimes GTP, that's why I call it the quantum of biological energy. This is, somewhat contextual, depends on the concentration, ionic concentrations, but somewhere in this range. So if you convert it into joules, which is the energy level in physics, it's roughly 10 to the minus 20th joules. So it's easy to remember. That's the quantum of biological energy. And we talked about quantum energy levels in physics, right? This is, to me, a close equivalent in biology. All of biology, you cannot use half ATP, you cannot use 3/4, you have to use one ATP molecule. One phosphate group detaches from this adenosine triphosphate, right? ATP. You have ATP, adenosine diphosphate. This one phosphate group carries the electrostatic energy, by the way. And that's the electrostatic energy that is imparted into, let's say, kinesin step, allowing kinesin to detach from actin filament and making it a 4 nanometer step forward, for example. So that is something to keep very much in mind. As you can see, it's clearly above thermal, this free energy available everywhere level. That means it has to be determined. And by the way, some people try to define what is life, including Schrödinger. To me, actually, a living system is a system that makes a decision, deliberate decision to move. Movement is essential, to move somewhere. And for this, you need to have energy, which is like this fuel, maybe I call ATP biological fuel, and then make a directional decision. Move towards the class end of a microtubule for kinesin, or or for myosin and actin. It's an act of deliberate action. Okay, so I'll stop here, and then on Thursday we'll actually get into the nitty-gritty. I will give two examples of quantum phenomena at the molecular level. One is photosynthesis, and the other is metabolism in mitochondria, mitochondrial metabolism, both of which speak to the energy requirement of living systems, both plants and animals. The most fundamental aspect of life, in my opinion, energy production and consumption, without which there is no life, there's no life processes, there's no quantum of energy. And then I'll go into a little bit more neuroscience-oriented discussions about where quantum physics may meet neuroscience or even consciousness, let's put it this way. Thank you. Questions, maybe. I'm sure there will be some questions. There's time for questions. 1 minute. So,
I have 3 questions, but I think we'll go over the time limit. I can just ask you after class.
okay, fine, I'll be here.
I have a quick question.
Yeah.
So you know how you briefly mentioned this, so just like I wanted to hear a little bit more detail if you could. You mentioned that the Bose Einstein Condensate, that it links somehow with the way that we think and the way that our thoughts
Well,
connect.
yes, because you see, it's a concept which is very attractive in it that it requires really no, it's a self-organized situation or self-induced process, which in physics, condensation would require lowering the temperature minimally above a certain threshold for the entire system to behave as one. And I think for the human brain, one of the very difficult questions to answer is, how is it that in spite of having 100 billion neurons, right, we have 100 billion neurons, each of which is connected to 10,000 to 100,000 other neurons by synapses, we still have this, think that this is you, this is me. I have my brain, and this brain understands itself as one, not 100 billion neurons. It's like chattering nonstop in an incoherent gibberish. So condensation would kind of provide a bootstrap to all of this. If they were somehow condensed into one state. By the way, Bose-Einstein condensation does not exclude fluctuations from it. That is, a condensate is majority of, let's say, a majority of neurons would be in the same state, but Bose-Einstein condensation allows other states also to be occupied, but gives you this framework where a majority is on the same wavelength. And it's called the two-fluid theory. So there's a condensate and normal part. Normal is classical, and then condensate is the quantum. So in other words, the single wave function for the entire macroscopic system, and that's the condensate wave function. So we'd like to have a wave function for your brain, for my brain, rather than, and we can talk about psychiatry, by the way, because there's something like, split personality disorders, right? Multiple personalities. People think that they have two different or more different voices talking to them inside the brain. Maybe this is some kind of breaking of the condensate. I take it as metaphors. Physics provides definitely very interesting metaphors for life sciences to understand, especially neuroscience. A very good question. Thank you.
So any other questions?
After class?
I got 3, but I don't know if you want to sit through it, to be honest. It might take a long, I don't know.
How about one? Okay.
One.
And the other two after.
Just really quick one then. So I know one way we can describe thermal energy is vibration. But then you talk about how there's a lattice and all of them are synced.
Yes. So
then vibration could be frequency but also amplitude. So how do both those concepts relate to thermal energy?
Yes. So the amplitude has to do with, you have to have, if you have a bigger amplitude, you have to have more energy. KT is the average, what you can expect. So if you increase temperature, these amplitudes will get bigger. But and maybe I'll step back and tell you a bit more about this since you asked about KT. So in statistical physics, there's this theorem called equipartition theorem. It tells us that every degree of freedom could be translational, so movement this way, that way, rotational, vibrational, at temperature T in that system has an average energy of KT divided by 2. 1/2 KT is the free energy, simply because the system is in the presence of something called the heat bath or thermostat. And that energy here is given by heating, right? So, and we also generate energy by burning, fuel and creating ATP and basically glucose metabolism. So KT divided by 2 is the energy that gets free because of the temperature provided to the system. To every so degree of freedom is that something, either rotational, vibrational, or translational motion that the system is not constrained by. So it can move. If you constrain it by, let's say, gluing somewhere or fixing it, then there is no degree of freedom. It is lost. In the case of lattice, you mentioned the lattice, these atoms don't have the translational degree of freedom. They cannot just escape typically unless you heat it up to vaporization temperature. They have the freedom to move around the equilibrium, and that's this amplitude. And the higher the temperature, the greater the amplitude.
So what was the frequency then?
The frequency, yes, as well.
So both of them are represented as heat.
Correct, by temperature, yes.
Okay, so we put more energy, we have like a bigger...
H omega basically, so Planck's quantization, H omega equals KT, or KT divided by 2, but the degree of freedom to be specific.
I guess macroscopically we also talk about like resonance frequency when that's called a constructive frequency. So what if we did that thing to a lattice, right? Normally you just put in more energy, which isn't resonance, right? But people resonance.
Yes. A good question too. I think for this we probably need a few minutes next time, or maybe after, because you're talking about something which is actually incredibly fundamental in physics. It's called the harmonic oscillator. And there's a classical harmonic oscillator and the quantum harmonic oscillator. And the quantum harmonic oscillator has these energy quantized levels. In the classical you don't. So you have amplitude is a continuum, right? It's a continuum variable. It can be anything. It's this pushing on the swing, that's the classical physics 101 example, where you have the kinetic energy going into the potential energy, it's basically spin constant K times A squared divided by 2 is the potential energy, and kinetic is mv squared divided by 2. So that's classical, but in quantum, you have a single frequency because it's quantization, and you have H omega divided by 2 times n. So you can have 1 quantum, 2 quantum, 2 quanta, 3 quanta, the same frequency. In 1 minute. Physics 101 and physics 471 or something.
Okay.
I still remember physics after not teaching it for 15 years.